What I'd like to talk about is the development and composition of biofilms on different oral surfaces. Now, the biofilms that form in the mouth are very complex. And if we look at supragingival plaque, which is a biofilm that we often see as clinicians when we look in a patient's mouth, and we take a sample of this plaque and look at it under the microscope, what we see is that there is a wide variety of different organisms that one can see in this particular biofilm. And what we can see here is different shapes of organisms. We have this long filamentous organism here. We have this rod-shaped organism here, this curved rod, these round organisms or cocci, these sort of corkscrew organisms which are spirochetes. And these different shapes or morphotypes actually represent literally dozens of different species. And in fact, we know that probably over 5 to 700 species can actually live in the oral cavity. It doesn't mean that everybody has 700 different species in their mouth, but the mouth is home for a very wide range of microorganisms. About 300 to 400 of these species can colonize the subgingival area. Many of these species have yet to be cultivated. We can grow approximately 50% of the microorganisms that live in the oral cavity. Species colonize anything in the mouth. They colonize the hard tissues, such as the teeth. They colonize the soft tissues. Anything you put into the mouth, they will also colonize, whether you put dentures or crowns or whatever in the mouth. These structures will become colonized by bacteria. Fortunately, most of the species that live in the mouth are host compatible. They are beneficial species. They don't do any harm. But a subset of the species are responsible for two of the most common infections of mankind, namely dental caries and periodontal diseases. And another important factor about these diseases is that the species that cause these diseases, dental caries and periodontal diseases, are endogenous species. These are species that naturally live in the mouth. They don't come from another source, come into the mouth and cause disease. They are already in the mouth. They live in the mouth. So when one's treating these infections, it's going to be much more difficult to eliminate. In fact, it will be impossible to eliminate these species from the mouth. So in our treatment strategies, we're not going to eliminate these particular species. The other thing that is important to note is that these species live in structures called biofilms, which we'll talk about in a little bit. But biofilms became prominent in the 1990s when it was recognized that infections on implantable devices such as hip replacements or indwelling catheters or heart valves were very, very difficult to treat. These devices became infected. The patients would be treated with massive doses of antibiotics, and nothing happened. The infections were not treated, were not controlled. And it was found that these species were living in these protected environments called biofilms rather than species living in what we call a planktonic state, sort of in a single-cell state on their own. It's also been estimated, and the Center for Disease Control, the CDC, has estimated that about 65% of human bacterial infections are caused by bacteria living in biofilms. And we have a few examples here. For example, cystic fibrosis and chronic sinusitis are caused by a species called Pseudomonas aeruginosa. Other infections, as I mentioned before, on medical devices such as the indwelling catheters, joint replacements, heart valves, are caused by species the Streptococci family and also Staphylococcus aureus. We have urinary tract infections, middle ear infections that are caused by bacteria living in biofilms. And, of course, periodontal diseases and dental caries are diseases caused by the species that live in supra- and subgingival plaque. So, we've talked a lot about biofilms. What are biofilms? Well, the official definition of a biofilm is that they are communities of microorganisms attached to a solid surface that are embedded in a glycocalyx. Now, what does this really mean? Well, if we look at a biofilm, there are three major components to the biofilm. First, there is the surface to which the bacteria attach. Then there are the microcolonies that grow out from this surface. And these microcolonies are embedded in something called a glycocalyx. And what a glycocalyx is, it's essentially water, but it contains exopolysaccharides which are secreted by the bacteria themselves. And this glycocalyx gives form to the biofilm. It also provides protection. Also, substances such as signaling devices can be concentrated in the glycocalyx, so it allows the bacteria within a biofilm to communicate with one another. We also find water channels in the biofilm, and these water channels allow the dissemination or the spread of nutrients among bacterial species in the biofilm. It also allows for waste materials to be removed from the biofilm. The third major component of the biofilm is the bulk fluid that actually bathes the biofilm. In the mouth, this biofilm is saliva, and what the bulk fluid does is it brings nutrients to the biofilm, and it also takes away the waste products, and it also allows for dissemination of species from this particular biofilm to another area so that they can colonize a new area, for example, in the mouth. Now, there are many advantages to species living in a biofilm. First of all, they are protected from competing species. Secondly, they are protected from host defense mechanisms which try to destroy and kill the bacteria. They're also protected from antibiotics. As I mentioned earlier, bacteria living in biofilms can be 500 to 1,000 times more resistant to antibiotics than bacteria living in a planktonic state. Living in a biofilm also facilitates the processing and uptake of nutrients. It allows for cross-feeding. For example, some bacteria produce certain substances like vitamin K, which is essential for the growth of other organisms. And living sort of in close proximity to species that produce substances that you need for your growth is a very advantageous way to live. It also allows for the removal of harmful metabolic products and also allows for the development of the appropriate physical chemical environment for the growth of the bacteria. It's almost like living in a city where you've got protection, you have the police, you have the fire department, you have highway systems, you have restaurants, you have shops where you can get food. So living in a biofilm is a little bit like living in a city for the bacteria, as opposed to living on the top of a mountain or by yourself where you don't have any sort of amenities at all. Now, the biofilms in the mouth that have been studied extensively are probably supra- and subgingival plaque. And a lot of work has been done on looking at the characteristics of subgingival plaque. And one of the studies that we performed back in the 1990s was to look at the association of bacteria within the biofilm. You may have noticed in the electron micrograph that we saw in the earlier slide that the organization of the species seemed to be somewhat haphazard. But that isn't the case. There are specific associations among bacteria in biofilms. And we looked at this particular issue in subgingival plaque samples. In this particular study, we looked at biofilm samples, subgingival plaque samples, from 185 subjects. We took subgingival plaque samples from every tooth in each of these subjects. And we analyzed these samples using checkerboard DNA hybridization. This is a molecular technique for the identification of bacterial species. Indeed, what one does is one identifies the DNA from the bacterial species in the plaque samples using whole genomic DNA probes. And then using that data and various statistical analyses, such as cluster analysis and community ordination, and these are basically techniques that look for associations among bacterial species, we found the following results. We noted that certain species were found more commonly together than with other species. So, for example, the Actinomyces species formed a group of organisms. The Streptococci species, which we call the yellow complex, formed another group of organisms. This green complex, which is made up of Eikenella corrodens, some Capnocytophaga, and Aggregatibacter Actinomycetum comitans serotype A, and a purple complex, which is made up of Veionella parvula and Actinomyces odontolyticus. Now, not only did we see these specific associations among bacterial species in these different complexes, we noted that these complexes were related to one another. And we felt that these were species that were early colonizers on the tooth surface, and they also were mostly species that are thought to be host compatible. These species set the stage for another group of organisms that we call the orange complex, which is made up mainly of Fusobacterium nucleatum species, Prevotella species, Campylobacter species, Eubacterium nodatum, Streptococcus constellatus. This orange complex sets the stage for what we call the red complex, which is made up of two periodontal pathogens, two official periodontal pathogens, Porphyromonas gingivalis and Tannerella forsythia, as well as Treponema denticola. And as we look at this scheme, we note that as we go from the left to the right, you go from more host-compatible species to more pathogenic species. As I mentioned, the species of the red complex are periodontal pathogens, and many of the species in the orange complex are also periodontal pathogens. This is just a very nice picture courtesy of Dr. Kuratmitsu, who looked at the relationship between P. gingivalis and T. denticola. And in this particular slide, we see the spirochete T. denticola, and here is P. gingivalis. So these are two of our red complex species, and this shows the actual physical relationship among these two particular species. Now, while we were looking in vivo at relationships among different bacterial species, because we were looking at plaque samples from individual subjects, Dr. Kohlenbrander at the National Institute for Craniofacial Research down in Washington was looking in vitro at the associations among pairs of different species. And what they found was that species that attached to the tooth surface were mainly species in the genus Streptococcus. Not only did the Streptococci attach to the tooth surface, they also attached or coaggregated with one another. The Streptococci set the stage for other species like the Actinomyces, Veionella species, and Dr. Kohlenbrander felt that the Fusobacterium nucleatum species, which we see here as this very large rod-shaped organism, was a bridging species. That these particular organisms were very important in linking the early colonizers with the later colonizers. And in his scheme, the later colonizers included species such as P. gingivalis, T. denticola, Prevotella intermedia. So one can see a lot of similarities between the scheme or the relationship of bacterial species to one another that we found looking at subgingival plaque samples from patients, and the similarity to the scheme that Kohlenbrander found looking at pairs of organisms in the laboratory. Now, if we look at the relationship of some of these complexes and species to the clinical status at the periodontal site, we can see differences as we look at different sites within the mouth. This slide is set up with the 40 species that we look at using the checkerboard DNA-DNA hybridization technique. The species have been ordered according to the complexes, so we have the actinomyces species, the purple, yellow, green, orange, and red complexes, and a group we called the other complexes, which were species that didn't fit into any of these complexes, or they are new probes that we have developed since we did the original study. And if we look at the microbial profile at sites that have pockets greater than or equal to 6 millimeters and that bled on probing, i.e. diseased sites, this is the microbial profile that one sees. One can see large levels or high levels of orange and red complex species. If we now look at the microbial profile of sites in the same subjects where the pocket depth was less than 4 millimeters, i.e. the so-called healthy sites in these diseased patients, we note that the profile is a little bit different. We have significantly lower levels of orange and red complex species. What is interesting is when you actually put the profile of healthy sites from healthy subjects on the same graph. And we note that the levels of species in the healthy sites from the healthy individuals are significantly lower than the healthy sites in the diseased individuals. So that's telling us that the healthy sites in diseased individuals are already different from healthy sites in healthy individuals. The other point of interest in this slide is that even in the healthy individuals, you see periodontal pathogens of the red and orange complex. And this is what I mentioned earlier. These are normal inhabitants of the mouth, and you do find them in health, but obviously they're at much lower levels and lower proportions than you find in diseased subjects. And obviously there are a lot of significant differences between species levels in the three profiles. One other factor that we have to remember is that even subjects who have the same disease, the same clinical condition, the microbial profiles might not be the same. These are microbial profiles from 6 subjects who were picked because they had very similar clinical conditions. They all had similar full mouth mean pocket depth, similar full mouth mean attachment level. They were of a similar age. They were all white females. And yet, you don't need me to tell you that these profiles do not look the same. Subject 5, for example, has very high levels of the orange and red complex species, and this is what we would expect from patients with chronic periodontitis. But the patient next door has very low levels of not only orange and red complex species, but all the species. Now, it could be that we are not detecting the species, the pathogens that are causing the disease in this patient, or this patient might be very susceptible to even low levels of pathogenic species. So this is something that one has to bear in mind, that you do see different microbial profiles even in subjects who have the same clinical characteristics. So from this part of the talk, we can conclude that there are specific bacterial associations that exist in oral biofilms. There is variability in biofilm composition from tooth to tooth and subject to subject. And the composition of biofilms is affected by the habitat. We saw pocket depth is a factor that will affect plaque composition. Now, one of the major features of a biofilm is the surface to which the bacteria initially colonize. And one of the questions we ask, does surface, if you have different surfaces, does that affect the biofilm composition? And in the mouth, we have many different surfaces. For example, we have hard tissues. We have the teeth. And we have other hard tissues that we as clinicians put into the mouth, such as dentures, crowns, orthodontic appliances. There are also many soft tissue surfaces, such as the tongue, the cheek, the hard palate, the epithelial lining of the periodontal pocket. These are all epithelial surfaces, but are actually quite different surfaces. We also in the mouth have different bulk fluids. We have saliva, which would be the bulk fluid to most of the biofilms of the mouth, but we also have gingival clavicular fluid, which would be the bulk fluid feeding and nurturing the subgingival environment. We should also remember that other factors also impact biofilm composition in the mouth, and I'm not going to talk about that today, but factors such as host-level factors, including diet, genetic background, systemic and local disease, various habits like smoking, will all have an impact on biofilm composition. Now, in the study that we were looking at the effect of different surfaces on biofilm composition, we looked at supragingival plaque samples from each of 36 periodontally healthy, 15 periodontitis, and 61 denture-wearing individuals. All the samples that we took were analyzed using checkerboard DNA hybridization. And if we look at the levels, the profiles in these three different groups of patients, we can see differences in the three groups. So, for example, we have the profile of the periodontitis patients, and you'll note that the orange and red complex species, the levels of these species in these particular periodontitis patients, are somewhat lower than the levels we saw in an earlier slide of periodontitis, and that's because these patients have been treated. So one has actually lowered the levels of pathogens in these particular species. But if we look at the profile of the healthy, we can see that the levels of the species are somewhat lower in health compared to periodontitis. This third profile is the profile of the supragingival biofilm on the denture teeth. And we notice that there are differences, and in particular, there are differences in the streptococci. The supragingival biofilms on the dentures have much higher levels and higher proportions of streptococci than the biofilms in the dentate individuals. In a second study, we looked at biofilms that developed on different surfaces in the oral cavity. And this was a study by Dr. Major that she completed a few years ago. We looked at samples on hard surfaces of supra and subgingival plaque. She also looked at biofilms that developed on different soft tissue surfaces, including 3 surfaces of the tongue dorsum, the lateral tongue, and the ventral tongue, the floor of the mouth, the hard palate, buccal mucosa, vestibule lip, and the attached gingiva. She also looked at the bacteria in saliva. And all of these samples, again, were analyzed using the checkerboard DNA hybridization technique. And these are the results. And this is a somewhat complicated slide. We have here on the left the species that were looked at. Here each panel represents a different area. This is supra and subgingival plaque. This is saliva, the tongue dorsum, the lateral tongue, the ventral tongue, floor of the mouth, buccal mucosa, hard palate, vestibule lip, and the attached gingiva. But once again, you don't need to know anything about microbiology of the mouth to sort of look at these profiles and say they don't look the same to me. And in fact, if we look at supra and subgingival plaque, we note that these particular biofilms are dominated by the Actinomyces species, and we don't actually see those species very much on the other surfaces. If we look at the red bars, which are V. parvula and P. melaninogenica, these species tend to be in the highest proportions in saliva, the tongue dorsum, and the tongue lateral, as we can see here and here, and are at lower proportions on the other sampled sites. If we look at the purple bars, which is G. morbillorum, Streptococcus mitis, and Strep oralis, these species tend to be higher on the other oral soft tissue surfaces rather than the lateral and dorsal surfaces of the tongue and the teeth. So there seem to be definite patterns of colonization among these different oral sites. And if one does a cluster analysis, which again is looking at associations among bacterial species, associations among the microbial profiles, we can see that the tongue dorsum, saliva, and the lateral tongue had very similar microbial profiles. The other soft tissue surfaces had also a very similar profile, and they were somewhat related to the tongue dorsum and saliva. And supra and subgingival plaque were somewhat different. They were similar to each other, but somewhat different from the other surfaces. So again, this is a simple way of showing that the microbial profiles on these different oral surfaces are quite different. If we now look at the same type of thing using our healthy periodontitis and denture patients, we can see that the profiles again differ among these groups of individuals. So in this particular slide, again, these are the species we looked at. Here we have saliva, tongue dorsum, tongue lateral, the ventral surface of the tongue, floor of the mouth, buccal mucosa, hard palate, vestibule lip, and the attached gingiva. And these are the profiles on these different surfaces of the healthy patients. These are the profiles of the periodontitis patients. And interestingly, there's not a lot of difference between the profiles. There are some differences. But in these two groups of individuals who both have teeth, obviously, the profiles on the soft tissues are not that different. If you look at the profile in the edentulous individuals, we note that the soft tissue microbiota in patients that don't have teeth is somewhat different from the subjects that do have teeth. And in particular, we have much higher levels of Streptococcus mitis on the soft tissues of the edentulous patients. And that might account for the fact why edentulous patients in their supragingival plaque on the dentures have high proportions of this particular species. And we note that other species like Eikenella corrodens and some of the fusobacteria are much higher in the dentate individuals than in the subjects without any teeth. And again, a lot of differences, significant differences among bacterial species in the three subject groups. Again, if we do a cluster analysis of these microbial profiles, as we did previously, we can see in the dentate subjects that the tongue and saliva profiles tended to cluster together. The other soft tissue tended to cluster together. In the edentulous patients, we see the same pattern. The tongue, the lateral and dorsum of the tongue profiles and saliva profiles cluster together, and the other soft tissues cluster together. So we see the same sort of pattern. But what is interesting is that there's absolutely no crossover between the profiles of the edentulous and the dentate individuals. So we can conclude from this part of the talk that surface is a major determinant of intraoral biofilm composition, and the oral soft tissues and saliva provide a source of bacteria for colonization and reinfection of the hard tissues. It also works in the other direction also. The next question we were interested in is there bacterial succession in biofilm development? And now we got a sense that this probably is the case looking at the microbial complexes work, that certain species seem to be early colonizers on the tooth surface. But we actually performed some studies to sort of see, how species come back. Because once you clean a tooth surface, not everything comes back at the same time. And in this first study, which was performed by Lee et al., they looked at supragingival plaque development over a very short period of time, over 6 hours. And what they did is they cleaned the teeth and took samples of plaque from those teeth. They then waited 2 hours for the plaque to develop and then took more samples. They then cleaned the teeth again, waited 4 hours, took some samples, cleaned the teeth again, waited 6 hours for plaque development and took some more samples. And what they found was that immediately after tooth cleaning, there are some bacteria still there. After 2 hours, the levels of species are somewhat higher. After 4 hours, the levels are higher still. And after 6 hours, the levels are higher still. Nothing surprising there. But what is interesting is that not all of the species came back at the same rate. And certain species, particularly Streptococcus mitis and Streptococcus oralis, came back much more rapidly during this short-term plaque development than other species. We also did a study where we were looking at somewhat longer-term plaque development over 7 days. And in this particular study, we took samples as the patient came in the door, as it were. We cleaned the teeth, and then we took samples immediately after tooth cleaning. And these were full mouth samples. We then took samples from quadrants, separate quadrants, at 1, 2, 4, and 7 days. And during this 7 days, we asked the patients not to brush their teeth or to clean their dentures, because the patients we're looking at, again, are these 36 healthy, the 15 periodontitis, and the 61 edentulous subjects. So we were looking at plaque development over 7 days in patients who were not brushing their teeth. And what we found was, if we look at some of the species and how they came back or how quickly they came back, this is what we see. So, for example, a species such as Strep mitis, this was to be the baseline value. It decreased in levels after cleaning, but by about 1 day, the levels were back to the baseline level. So this particular species came back quite quickly. Fusobacterium nucleatum, subspecies nucleatum, again decreased after cleaning and gradually increased and was probably back to baseline levels at about 4 days. Prevotella intermedia took a little bit longer, somewhere between 4 and 7 days. It came back to baseline levels. And the periodontal pathogen, T. forsythia, actually did not reach baseline levels during the 7 days of this study. So it's quite clear that species come back at different rates in plaque. If we look at the proportions of these particular species, how much did they make up of the total plaque mass? We note that the Streptococcus, the Strep mitis, was a very high proportion of the plaque early on, but then declined, whereas the Prevotella and the Fusobacterium were in low proportions to begin with, but then gradually increased in proportions over time. Again, Tannerella forsythia did not reach particularly high proportions during the course of this particular study, but we know that's going to change because these particular species in diseased people are in quite high levels and proportions. So clearly something else happens after 7 days of plaque development. The other thing we were interested in, noting our results and noting the results of Lee et al., is whether patients who had different levels of streptococci at the beginning of the study, whether this impacted the rate at which the plaque returned. So what we did is we subset patients into those that had low levels of Streptococcus mitis and oralis to begin with. They naturally didn't have very high levels of these species. And subjects who had moderate or high levels of these species. And then we looked at the impact on plaque development in these particular subjects. So we looked at the effect on total bacterial counts, on counts of Actinomyces niselandi 2, Strep sanguinus, and P. gingivalis. This yellow profile represents the subjects who initially had low levels of streptococcal species. And what we see is that the species went down after cleaning and increased. But in all cases, the levels didn't reach or reached baseline levels or barely reached baseline levels at the end of the study at 7 days. However, in subjects that had medium or high levels of streptococci to begin with, we note that the species came back much more quickly, and at the end of the study were in much, much higher levels than they were at baseline, again suggesting that the importance of the streptococci as initial colonizers on the tooth surface and their effect on plaque development or subsequent plaque development. So we can conclude from this part of the study that there is microbial succession in development of biofilms. Pioneer species such as Strep mitis and Strep oralis increase in proportions within 2 to 6 hours, as we saw in the Lee study, and peak at about 1 to 2 days and are necessary for plaque regrowth. Other species, such as P. gingivalis and T. forsythia, the periodontal pathogens, increase more slowly, which is fortuitous for us as clinicians. The next question is, does surface affect microbial succession during biofilm development? We saw that surface did have an impact on the composition of the plaque, but does it have an effect on the rate at which plaque redevelops? And in this particular study, again, we're looking at the subjects with periodontal health, the periodontitis, and the edentulous subjects, and we're looking at the 7-day study from baseline to 7 days. And we're looking at the total bacterial counts in this particular slide. These are the baseline levels of the bacteria in the 3 groups. In health, somewhat higher in the periodontitis and somewhat lower in the edentulous, at least compared to periodontitis. In the healthy individuals, we notice after cleaning the counts go down, but by 2 days the counts are beyond the baseline levels. Same sort of pattern in the periodontitis patients. Again, by 2 days we have reached pretty much the baseline levels of plaque in those individuals. In the edentulous subjects, the picture is a little bit different, and it takes about 4 days for the counts, the total counts of bacteria, to reach the baseline levels, suggesting that surface does have an effect on biofilm redevelopment. If we look at the full set of bacteria, again looking in the health, the periodontitis and the edentulous subjects, we can see how all of the species came back over time. Here we have the baseline profiles immediately after cleaning. This is at 1 day, and this is at 2 days. And we note that in the healthy and the periodontitis subjects, this yellow profile has already pretty well covered the green profile, which was the baseline counts of the species. So again, by 2 days in the dentate subjects, most of the species have reached the baseline counts. Whereas in the edentulous subjects, this is definitely not the case. And it actually takes 4 days, when we look at this orange profile, for the species, the counts of most of the species, to reach the baseline levels. And this is just the 7-day profile. And if we look specifically at examples of bacterial species, we're looking in the healthy individuals and the edentulous, we can again see that Strep mitis comes back much more quickly in the dentate, healthy individuals than the edentulous. Same is true for Fusobacterium, same is true for P. gingivalis, same is true for Actinomyces nislandi genospecies too. And the reason for this is probably, A, we have a different surface. We're talking about a plastic denture versus a tooth. But we're also, must realize that in the edentulous subject, there's no subgingival environment. And the subgingival environment probably does help to feed the bacteria in the supragingival plaque. You have GC, gingival crevicular fluid, that also helps and provides nutrients to the supragingival environment. So that's probably why one sees this delay in the growth of species in the edentulous subjects. So we can conclude that surface does have a major impact on bacterial succession. Now, the final section of this talk is, can we actually affect? Can we change these biofilms? And as clinicians, this is basically what we want to do. When we treat patients, we want to be able to change pathogenic biofilms to one that are more consistent with health. And I'm going to show 3 different treatment effects in the next few slides. In this first study, we're looking at the edentulous subjects. We're looking at the effect of soft tissue disinfection on biofilm development on the tongue. And what we did in these patients is we cleaned the tongue, we brushed it with chlorhexidine, we soaked swabs in chlorhexidine and wiped the soft tissues. We had the patient gargle and rinse with chlorhexidine. And we looked at the effect of plaque redevelopment on the tongue in the edentulous patients. So again, we have the baseline to 7 days. And in half of the subjects, this soft tissue disinfection was not performed. And what we see is that over the course of the study, there really wasn't much change in the counts of the species in these particular biofilms. In the patients who had full mouth disinfection, we note that at 2 days, or at day 0 and day 1, there was a significant decrease in the counts of the species in the subjects who received the soft tissue disinfection. By 2 days, there was still a bit of a difference, but this was not significant, and by 4 days, there was no difference. But what we're seeing is that by cleaning the tongue and the soft tissues, this did have a major impact. This one-time cleaning had a major impact on the redevelopment of the plaque on the tongue. In a similar experiment of disinfection, we're looking at the effect of soft tissue disinfection on plaque development on the teeth. So here we're looking at the periodontally healthy subjects, and we're looking at supragingival plaque redevelopment. So once again, the subjects received the same soft tissue disinfection, brushing of the tongue, cleaning of the soft tissues with a gauze soaked in chlorhexidine, gargling, rinsing with chlorhexidine. And this is the group of subjects that did not receive the soft tissue disinfection. So we see the pattern, because they obviously had their teeth cleaned, and we see the pattern we've seen before of plaque redevelopment over the 7 days of the study. In the subjects who received soft tissue disinfection, we note that at 1 day and 2 days post-disinfection, that there was significantly less total plaque on the teeth in the subjects who received the soft tissue disinfection. Again, by 4 days, this difference had disappeared, but this one-time soft tissue cleaning did have an impact on the rate of plaque development on the teeth. What about some of our periodontal therapies? This is a study in which we looked at patients who had received various periodontal therapies, surgery, scaling and root planing, but also received systemic antibiotics. Here we have the profile of the subjects at baseline, and this is the profile of the same subjects at 12 months. And we can see that there have been significant reductions in many of the species, particularly members of the orange and red complexes, the pathogens that cause these diseases. So clearly, one can, even 12 months after the treatment, one has a major impact on the subgingival microbiota in these particular subjects. So I think we can conclude from that that we can actually affect the biofilms in the mouth. We can change the composition by our therapeutic interventions. Now, you might be asking yourself, why do I need to know any of this? Well, I think one thing is, as individuals, clinicians who work in the oral cavity, I think it's important that we understand the microbiology, the ecology of the mouth, so that we understand what we are doing or how we affect the microbiota or the ecosystem in the mouth when we do our treatments. But there are other considerations, and whether it's you as a clinician designing treatment planning or treatments for your subject, or whether you're a company designing toothpastes or mouthwashes to try and prevent and treat diseases, there are certain things that you should remember. First of all, there is an interplay between the biofilms that colonize the teeth and the soft tissues. So maybe it's not enough just to treat the teeth. Maybe we should also be treating the soft tissues, because the soft tissues are reservoirs of species, including pathogens, that can reinfect, recolonize the teeth once you have treated them. Secondly, pioneer species are critical for plaque development. We saw that the streptococcal species in particular were particularly important for plaque development. If you had low levels of these species, plaque developed much more slowly in those particular